What if I told you we could edit life itself? I'm not talking about some sci-fi movie, I mean right now, in a lab. Today we're going to dive into one of the most incredible tools ever invented, the power to literally rewrite our own genetic code. So let's get right into it. I want you to imagine the human genome as this enormous instruction manual. We're talking 3 billion letters long. It's the blueprint for, well, for you. But, just like any book, sometimes there's a typo. A single wrong letter that can change absolutely everything.
A way to go into that massive book, find the exact misspelled word, and just correct it. Precisely. Permanently. That is the incredible promise of gene editing, and the story of how we're getting there is honestly just fascinating.
So, our story really kicks off with a technology you've probably heard of, right? CRISPR-Cas9. This was the tool that truly blew the doors open for widespread gene editing.
The concept behind it is actually elegantly simple. First, you have something called a guide RNA that acts like a GPS, leading this protein, Cas9, to a very specific spot in the genome. And once it gets there, Cas9 does one thing and one thing only. It cuts. It works just like a pair of molecular scissors, making a clean break right through both strands of the DNA. Then, the cell itself kind of panics and rushes in to try and repair that break.
And right there, you can see both the power and the problem. On one hand, yeah, CRISPR is a revolutionary tool. But on the other hand, that double-strand break, it's a blunt instrument. It can really stress out the cell, and that repair process, it's often messy, it's unpredictable, and sometimes it even creates brand new errors. So the takeaway is pretty simple. CRISPR is amazing at breaking genes, but it's not so great when you need to precisely fix them.
So, this need for more precision led to the first major evolution in gene editing. Scientists started asking, okay, what if we could edit, but without making that dangerous cut? And that brings us to base editing.
Instead of molecular scissors, I want you to think of base editing as a pencil with a very, very precise eraser. It still uses a modified Cas9 to find the right spot in the DNA, but instead of cutting, it brings along an enzyme that chemically changes one DNA letter, one base, directly into another. No cut, no break, just a clean, simple chemical switch.
And this was a huge leap forward. I mean, the pros are pretty obvious. No more of those dangerous double-strand breaks, which makes it super efficient for fixing those single-letter typos. But, and this is a big but, look at the con side. This is its Achilles heel. Base editing is like a specialized tool that can only do one or two very specific jobs, like changing a C to a T. But what about all the other kinds of typos?
And that limitation, well, that brings us to the true star of our show today, prime editing. If CRISPR is like scissors and base editing is like an eraser, then I want you to think of prime editing as the search and replace function on your computer.
The way this works is it's pure genius. It uses a modified Cas9 to just gently nick one strand of the DNA, so no dangerous double cut. Then it brings in a second enzyme, a reverse transcriptase that acts like a molecular printer. It uses an RNA template to directly write the correct genetic sequence right into that spot. It doesn't just cut or erase, it finds the typo and replaces it with the right text, all in one go.
So why is this such a big deal? Well, the advantages are massive. First off, just like base editing, it's way safer because it avoids those double-strand breaks. But here's the real game changer, its versatility. It can correct all 12 types of single-letter mistakes, and it can precisely add or even remove small chunks of DNA. This isn't just a small improvement, it's a gigantic leap in what we thought was possible.
But you know how science is, right? It never just stands still. Ever since it was invented, researchers have been working non-stop to make this incredible tool even better. The pace of innovation here is it's breathtaking. This timeline really tells the story. Back in 2019, the first version, PE1, just proved the search and replace idea could work. A couple years later, by 2021, PE4 and PE5 came out, and they dramatically boosted efficiency by basically telling the cell's own proofreading system to take a quick coffee break, which gives the edit time to stick. And then, just this year, we got PE7. It adds a protein that acts like a little shield for the guide RNA, protecting it and making the whole process even more efficient and accurate.
Okay, this is all amazing science, but it raises the most important question of all, right? What can it actually do for people? Let's take a look at how prime editing is finally making that incredible jump from the lab into our lives.
This quote from just earlier this year marks a truly historic milestone.
Prime editing isn't some theoretical tool we hope to use one day. It has officially been used in human patients for the very first time, and the initial results are showing that it's both safe and effective.
The very first published data comes from a clinical trial for a rare, devastating genetic disorder called Chronic Granulomatous Disease, or CGD. It basically cripples a person's immune system. In 2 patients, scientists used prime editing to correct the faulty gene in their blood stem cells, and it appears to have worked.
And what's so cool is that the potential applications are incredibly broad. On one side, you've got medicine, where it's being used to treat these terrible genetic diseases like CGD. But on the other side, you have agriculture. In places like Rwanda, scientists are now using these same tools to develop crops like potatoes and cassava that are more resilient to climate change, directly helping with food security.
Which leaves us with this one powerful question. For the first time in the history of life on Earth, we have the tools to correct the typos in the Book of Life. The science is no longer the barrier. The only real question left is, what will we choose to edit next?
Thanks so much for spending some of your time with us today. We build these videos for you, so your feedback is our compass. What's one subject you've been wanting to see a deep dive on? Let us know in the comments section. We read through your suggestions to decide what we film next. Don't forget to subscribe and turn on notifications so you don't miss our next upload. Stay curious, and we'll talk to you in the comments.